Hello, everyone, and welcome to today's Troubleshooting GenBank Submissions webinar. Our focus today is on eukaryotic genes. My name is Bonnie Madak. I'm the presenter of today's webinar. My colleague, Maida Valovets-Gratian, and I put together this webinar to help you with your data submission. We are part of the NCBI General Help Desk team that are trained to assist data submitters. We also have with us today Michael Fetchko, who is a GenBank staff member who processes many of the data submissions. We also have with us Peter Cooper, who heads the outreach, the educational component for NCBI activities. All of us can be reached via the info@ncbi.nlm.nih.gov email address. The slides and the sequence files that we're going to be using today is at the FTP path that you see there on the screen. We have also shortened the link to be this other usa.gov, that is 1.usa.gov/1 capital H Y X capital F capital E A link. And then all of the content from today's webinar and a video recording will be available probably within a week or so after the webinar is over. On the slides, you will see bracket NCBI bracket, and you need to replace that with the full NCBI URL as I show on the screen. We will respond to questions as we go, but all of the questions will be posted online after the webinar recording is available. So our goals for today's webinar is that you're going to be able to read the feature annotation in a GenBank record and understand how it will help you for your data submission. We're also going to tell you how to form the feature annotation, how to structure that information in the submission tool of choice. And we're going to be using BLAST as an analysis tool to determine the coding region locations on genomic DNA sequences that you will be submitting from eukaryotic protein coding genes. You're also going to receive some information to help you determine which GenBank tool is a better match for your data set. And because we're really focusing on things outside of the GenBank submission tools, this content is appropriate for anyone who is submitting, regardless of using Bankit or Sequin. Our outline today is as follows. There will be an introduction. Then we'll go through part 1, which is examples of eukaryotic protein coding genes. We're going to be using BLAST for 3 different data sets. And then part 3 will be actually showing you some of the steps that are needed for adding CDS annotation. And then the 4th part of the webinar will be going through some troubleshooting and error reports. So for the introduction, it's important that you know these biological concepts. We covered these in the previous Troubleshooting GenBank Submissions webinar, and I have the YouTube link there for the previous webinar, and the specific time points of that webinar are listed on the screen, around the 4-minute mark to the 22-minute mark of that webinar video. Our new biological concept for today's webinar is the structure of a nuclear eukaryotic gene. And the fact is that many eukaryotic genes, not all, but many, are broken up in the sense of the parts that comprise the messenger RNA, or they're not continuous in the DNA sequence. So a eukaryotic gene that is on the genomic DNA sequence has different parts, specifically exons, that go into the messenger RNA, but the introns are spliced out of the messenger RNA. The messenger RNA is then read by the ribosome to then be translated into protein with the amino acid residues. You might already know why you are choosing your specific gene that you are sequencing, and I've just put this material together so that you can have perhaps an additional sense of why other people sequence their genes. Some of the common eukaryotic molecular markers will be shown on the next few slides. For plants, it's really chloroplast genes that people commonly study, specifically the RBCL gene, which is an intronless gene. There's also a lot of people who study the MATK gene, and then there are regions such as the PSBA transfer RNA-H intergenic spacer region. For fungi, many people study the internal transcribed spacer region. That is referring to this part which is underlined with the red line, but that is part of the ribosomal RNA cistron, which includes the small subunit, the two internal transcribed spacer regions, along with the 5.8S and the 28S, or the large subunit ribosomal RNA gene. I want to point out that there is no such thing as the ITS gene. It is internal transcribed spacer, and your feature that you're going to need to add for that is a MISC RNA. That's MISC_RNA. Sometimes submitters call it a gene, and that is incorrect. Three other genes that are often studied in fungi are beta-tubulin, translation elongation factor 1 alpha, and then actin. And our example later in the webinar will be beta-tubulin. In animals, many times it is the mitochondrial genome genes that are studied as markers. The cytochrome C oxidase subunit 1, CO1, has other designations as shown. This again is an intronless gene because it is in the mitochondrion genome. There are other mitochondrial genes that are often studied. Another one is cytochrome B. And then another gene that is often studied is from the nuclear genome, and that is RAG1. So your goal is to get from the sequence on your computer to a GenBank accession number. And that's actually a misstatement, because what we really want for you to get a sense of today is that it's not just an accession number that you need to get. You want to have an accurate and complete GenBank record. It does not help you or any other data submitters to have just an accession number that does not correspond to an accurate and complete GenBank record. And what we're going to do today is, from a GenBank record, we're going to help you learn how to do the analysis and annotation of your sequence. Understanding the GenBank record display will help you prepare your data submission.